What if I tell you that in lakes and oceans you can find an important piece to understand the puzzle of neural diseases? So, nowadays, neural diseases as Parkinson or Alzheimer are in the high priority for scientists to solve or even understand how they work. However, the current, let's say, the current treatment or even the drugs that we are using are not enough, or even the successful rate is not enough for the scientists and for sure not for the patient itself. So I think we have a problem with the slide because it's not passing through. This is technical problem, don't worry. Okay, good. It's moving or not? Good. And all these problems that we are facing nowadays, what will be happen if I tell you that there is an upcoming technique that is less invasive and can solve all these problems, or at least we can study? And even what happens if I tell you that we need a specific type of proteins that I am in charge to find and characterize? And even more, what happens if I tell you that you can find those proteins in the most common places on the earth just using a very simple technique? Sounds amazing, right? So right now, what we are gonna do is try to follow the history and solve the puzzle together. Anyway, yeah, okay. We discovered the problem. So let's move on. Good. Now I need your help. I want you to let me know what these two pictures have in common. And I am not trying to sell you a vacational package for this summer. I'm pretty sure that many of you know the answer. That is water. That's right. But I'm gonna try to convince you that there is another possible answer for that question. There are also invisible elements that these two pictures have in common, but not all of us are aware of its presence. I am talking about microbes. My name is María del Carmen Marín. I am working as postdoctoral research at Technion, and my job is currently understand how those microbes interact with the environment, with other microbes, and with other creatures. But in science, we are facing many problems, and there are a lot of mysteries that we want to understand or even solve. Let me tell you the problem that we are facing in my field nowadays. 98% of those microbes that are found in marine system or freshwater lake are not able to grow in pure culture. What does it mean, pure culture, in microbiology? That means that they are not able to grow by their cell, like single organisms, in a laboratory plate. So here we have a strong problem that we need to overcome. Or we move our laboratories to those places, or we try to find a solution to overcome the problem. So this is why we will use environmental genomics. We will use environmental genomics as a tool to solve this problem. Genomics combines biology, genetic, and computer science to have a look in deep of the DNA. All living things in the environmental space, let's say from the most simple creature like single cells, microbes, to plant, animal, and human, contain DNA. So what we can do, using genomics, we can search for those DNAs. Each living thing in the environmental space has a unique genome. When we talk about genome, it's just the fancy word that we use to talk about DNA. And as research, we can use this DNA to look for the organisms that are in a specific place, like if we are looking for drop pins or for footprints, like with microbes. And easily we can know if a sample of DNA is containing in a bottle of steam water or in a cup of dirt, okay? How we'll do that? In an easy way. We know the amazing potential of the environmental DNA. This is the DNA that can be extracted from single substrate like soil or water, right? You need to know that each living thing on the earth, it's leaving a trail of breadcrumb of the DNA that contain like hair, urine, saliva, feces. And us, as researchers, what we do is, following that DNA, we press the rewind button and we can localize which organism passes recently from a specific area. So it's like, since each living thing has a unique genome, even if between species they contain broadly similar DNA, there are unique differences that make this species unique, and we can discover if it is there. So how we correlate this idea of environmental genomics with the important tool that we know, but that we need to solve the problem of neural diseases? If we look into the genome of microbes, we will find genes. Genes contain the hereditary material. For example, in humans, genes contain the information about the color or the hair color, the eyes color or the hair color, okay? So we do exactly the same on microbes. We look for the genes and then we evaluate the proteins that they are producing. So now that I introduced to you the word protein, I would like to present a very special kind of protein that I am in charge to study and discover, and why they are so important for us. I am talking about microbial rhodopsin or rhodopsin proteins. These rhodopsin proteins are light-sensitive proteins that use light to do activities in microbes, like, for example, transport nutrients across the membrane, okay? And I want to tell you that in this room, right now, there are rhodopsin proteins. I want you to look into the eyes of your colleague that are sit with you in the table, and you will be able to do it because rhodopsin are in charge of the vision process of humans. But regardless of the proteins that I am talking right now, and even the loving couple that can be generating after this intense looking on the eyes, we will talk from now about microbial rhodopsin proteins that are the rhodopsin proteins discovered in microbes. And for what microbes use rhodopsin proteins? They use it for harvesting light energy. So it's the most simple way in which the microbe use rhodopsin to take the energy from the light and make some cellular process. Of course, there are many microbial rhodopsin, as you can see in the slide. But for us, what are most interesting, because its application, are those ones that are able to pump different ions like calcium, sodium, potassium, chlorine. All these ions that easily you can find in food or when you are drinking milk, say extra calcium, okay? And they are so important because the mechanism is so simple. They receive the light, they open the membrane, all these ions are passing through the membrane, and then we create an ion motif, so like a channel in which are moving all these ions. And why is so important? Because if we search deeply in ocean and lake, we discover more of the rhodopsin that are already found. We can apply in a technique called optogenetic. And you will ask me, Maria, optogenetic is a very strange word. What does it mean, optogenetic? Well, optogenetic is a technique that uses the light to control cells, mainly neurons, in living tissues. So how can we connect microbial rhodopsin and optogenetic technique? Easily. What we are gonna do is take those microbial rhodopsin, we are gonna introduce into the membrane, and researchers will be able to activate that protein using light. So when they use the light, you will see that we will create a channel of ions and we will activate that neuron. Let me show you an example because with picture and videos it's much easier. For example, here you see a fly, okay? This fly in 2 of 200,000 neurons contain a light-sensitive protein. When the fly see a reflect of light, you see that they start to jump and try to fly. Of course, it cannot do it because it is trapped in a petri dish. What we are doing with the light is open the activity of the neural motor that make the fly flee. Like if we try to swap it, the fly will fly, right? So what we do with the light is exactly the same. But we control when the fly jump or spread the wings. Of course, we are not only interested to activate those neurons, we are also interested to silence them by switching off the neurons. How we do it? In the opposite way. We use a different microbial rhodopsin using different color of light, so we create this time a different ion path of ions, this time negative, and we can do exactly the same but with a worm. For example, now you can see a worm work out, moving, swimming, but when the light, the yellow light, is activated, the worm is stopped, not moving, okay? This is because in the neuron of the worm, we have a light-sensitive protein that when we irradiate with that light, we inhibit the motion of the worm, so this is why the movement stops. So we already learned that environmental genomics is the tool of the future that we are using today to discover novel microbial rhodopsin, which applications in optogenetic are endless. Research have already used the light to control the stumbling of a rat with Parkinson's disease and convert it in a steady wall. So, what will be next? It was a pleasure to be here to you presenting the basis of my research. And of course, I invite all of you to discuss with me your questions and curiosities during the Q&A session. Thanks to the Azrieli Foundation for giving me this opportunity, and thanks for your kind attention.
Questions? My name is Monica. I'm from Weizmann Institute. Really beautiful presentation, very interesting. I was wondering why you're looking at an environmental genomic sample, because I assume there are already a lot of rhodopsins from bacteria or microbes studied. What is the advantage looking at so many different? Are they special in terms of when you want to use them later, or you hope to find special features there?
So if I understand correctly, you are asking me why we decide to use environmental genomic in this specific field and in microbes and not in bacteria or other one. Yeah, that's true. This is because the rhodopsin that are found in microbial rhodopsin in microbes contain this specific characteristics that it's so important for us in optogenetic. They have this simple mechanism that using the light from the sun open the mechanism of pumping ions through the membrane and easily can apply in optogenetic. Perhaps there are other groups that are working with bacteria, but in my field, I am just focused on microbial rhodopsin.
Thank you.
You're welcome.
Hi, so my name is Carlos Bravo from Hebrew University of Jerusalem. And my question is, you mentioned during your presentation that one application could be potentially helping cure, in a way, or treat Parkinson's disease. Mm-hmm. Do you have any other examples of applications of these kinds of techniques that you're talking about?
That is a good question. So I just presented the most simple application. For sure, not only in this single species like a fly or a worm, but already there are a lot of studies on mammalian, like a rat mainly. But unfortunately, since this is not the field in which I am focused my research, perhaps I don't have enough information to answer that question. But for sure, there are other applications related with optogenetic. That is true that they are also using light for studying just tissues and other kind of organs that are relative to humans.
Raphael from beautiful Barilan. In the context of the pathology, I was wondering, how do you think we can develop such therapeutic, because it will require transgenesis, so modification probably, or maybe we can, I don't know, if you have any idea, like how to inject this protein in the patients.
Okay, so your point is the process, how we apply optogenetic in the patient. So for example, if we think in rat, what they do is use this rhodopsin, this protein itself. They introduce in a virus and they infect the neurons in the brain of the rat, and this is how they introduce the protein that later will be excited or inhibited with a specific type of light.
Yeah, I understand. In terms of ethics, it will require you as a patient to accept that you will have a kind of invasive genetic techniques on you.
No. Right now, for example, this technique has 10, 12 years. It's very young technique, but the first test required that the rat contain a fixed laser on the brain. So you see a rat moving connected to a network with blue light. But the purpose, and this is what I am mainly focused, is when we search for microbial rhodopsin, we are interested that they absorb light in a specific frequency of the spectrum, mainly in the infrared spectrum. Because if they do so, they only need to radiate the patients with infrared light that is not damaging the tissues or the organ that you are using. Unfortunately, the blue light that they're using nowadays, yeah, there are certain damage on neurons and cells, so this is why, mainly during this project, I would like to find those specific proteins that can avoid the damage of the cells.
And last question, like,
yep.
do you know infrared, how deep it can go through the tissue? Like, if we want to reach like central of the brain, it will...
No, I'm not sure. I know that go deep to the tissue, like maybe 3-5 millimeter, but I'm not totally sure. I know that as much we find protein in that infrared spectrum. For example, now the most redshifted protein that we have is cryptosome that absorb around 638, if not wrong, but it's the most redshifted. So the purpose is, even if we have natural protein that absorb in that spectra, make mutations that can even do more.
So it will be like very far red.
Exactly.
Okay, thank you.
You're welcome. Thank you.